the last part of chapter 17 that is related to hemostasis and also to blood types. And then we're going to start with chapter 18 with the heart, right? And I want to remind you that the topics are quite dense. There is quite a lot of memorization required. And yeah, sometimes it's a pain in the neck because there are so many moving parts and we tend to get lost in the middle of all this information. So I will advise you to go to SI sessions if you did not start going to SI sessions yet. Okay? And make sure that you ask questions and be on top of the topics because it's a lot. It's a lot to digest. And also, I want to encourage you to use all the resources that are at your disposal. For example, AI tools, right? Make sure you wisely use your AI tools like ChatGPT. Right? How many people have been using ChatGPT to study? Let me see, like show of hands. Yeah. Do you ask questions for the thing and you expect answers, or do you put prompts up and you say, write, I don't know, 50 multiple choice questions based on this topic or something like that? Hopefully, yeah. Who is familiar with Notebook LM? One person, two people. All right, honestly, I want to see hands up. Who is familiar with Notebook LM?
I'm not.
No, okay, who is not familiar with Notebook LM? All right, you're gonna like this tool, okay? If you want to stay a little bit after class, I'll give you a 5 minutes intro to that Notebook LM, or look it up on your own. You're really gonna like it, okay? All right, so here, let's dive here into our, yes, you have a question? Yes, usually for my exams, for the unit exams, it's, since this is the first exam, it's from where we started until the lecture before the exam, until whatever I get covered till then, okay? And I have it set in a certain way in the syllabus, but if we don't cover all the topics that I'm planning to, then I don't just put things in the exam and hold you responsible for it. I like to make sure that I cover all the things that I think are important just before the exam, okay? So it's from where we left off until what we get to. All right. Okay. Any more questions about the exam? When is the exam? February 2nd, here. Okay. All right, so we left last time talking about coagulation, right? And we see there are certain parts, certain stages to the coagulation, right? I had left off in this slide over here. So basically, the first thing that happens is a little tear in a blood vessel. And when you do have a tear in a blood vessel, you're exposing fibers, like collagen fibers, from the outside part of the blood vessel that usually the blood does not get exposed to, right? So what happens is that the factors that are in a zymogen form in the blood being not activated, when they get exposed to those factors like collagen factor or to outside factors, those zymogen factors get activated, and then you start on this cascade of first you're going to activate your platelets, and then you are going to activate the coagulation cascade. So there are many steps that happen, and there are many proteins involved in this cascade. A couple of things that is really important to remember is that the activation of the coagulation cascade is one of the two examples that we have of positive feedback. Okay, so triggering the cascade will make response bigger and bigger and bigger. And what we have seen before in this class, especially in the AP1 class, it was examples of negative feedback. When you take something out of balance, usually the body will counteract that thing to bring things back to normal, right? But in case of coagulation, you don't have the negative feedback. You have a positive feedback. You start the trigger, and the thing gets bigger, bigger, and bigger, right? Because you really need to form a plug. You need to stop the bleeding at the blood vessel. So first thing that happened was the platelet plug, right? You remember there were some factors that platelets release when they get activated, right? First, they are like a kind of little round little cell fragment, and once they bind to von Willebrand factor, VWF factor, the little tear in the blood vessel, then the platelets will get activated. They're going to become like star-like. They're going to have little spikes, and they're going to start to aggregate. They're going to call more aggregation of more platelets. When the platelets get aggregated, they are going to release three important factors. Actually, I like to remember two of them. One is ADP, and another one is serotonin, right? And there is another one, prostaglandin something. But I like to remember those two factors. And those are going to recruit more platelets to go to the site and work on making that platelet plug. Okay, all of those processes are a positive feedback type of thing. Right. Then after the platelet plug is there, this is when you are going to come with your coagulation. These are stronger. This is a stronger plug that is going to be on top of the platelet plug. And what you're going to do is you're going to form this gel-like substance from the blood components, and you're going to activate some fibrin threads to go on top of the platelet plug and make it really a strong cap, so that blood stops to flow through that hole. Okay, so what is involved here? You have here this enzyme called thrombin. Okay, so thrombin is going to convert a precursor of that fiber. So before it gets activated, that fiber precursor is called fibrinogen. Okay, so here is fibrinogen. So in order for you to get fiber, you have to get the fibrinogen and activate it. And the enzyme that does that is called thrombin. Okay, thrombin breaks fibrinogen into fibrin threads. Okay. All right, so we're forming the clot. Now, this whole process of getting to the fibrin involves about 30 or plus factors, right? Some proteins that are floating around in a very peaceful form and not activated. Those are the gen guys, right? But once they get activated, they start to bump into each other and get more and more activation. And those are called your clotting factors, right? So in average, we have clotting factors that are in Roman numerals. So they go like from 1 to 13, and you have other factors that are also playing around here, okay? Most of these factors, as we mentioned, they are proteins, and they're floating around in the blood. And what produces the proteins is the liver, okay? So liver is producing those clotting factors. Now, several of the clotting factors, in order for them to work properly, they need vitamin K, right? Now, we do not produce vitamin K. We have to recycle usually the vitamin K that we have. Remember, vitamin K is lipid-soluble vitamin. Do you remember all of your lipid-soluble vitamins? Yes? I heard A, D, E, K. Okay, so everybody remember, right? A. I like to think in this other order. I like to remember A, K, E, D, because vitamin ends with N. You put a little N in there, and then you get naked. And now we cannot forget them. Ha! Those are your lipid-soluble vitamins, right? So K is one of them. We do not produce it. We depend on our friendly microbiome, and we also depend on ingestion of some certain foods in order to produce your vitamin K. Very, very important for making some clotting factors. I'm not going to bore you with memorizing which clotting factors those are, but I want you to remember some important things about vitamin K, right? Okay, first thing, it is important. If you don't have it, then you do not have proper coagulation, all right? Now, there are some anticoagulants that will actually prevent coagulation from happening, right? Because that's the whole meaning for anticoagulant, right? So this factor here, antithrombin, is present in the blood and is usually active to prevent coagulation from happening when you don't really need coagulation, right? So there are certain things that are going to prevent coagulation from just happening. And the coagulation cascade can really happen anywhere, anytime, if you do not have proteins or factors that control this cascade. Okay, so here I give you an example of antithrombin that is an important checkpoint to avoid spontaneous coagulation. Okay. So here is a summary of events of hemostasis, right? And this panel here I took from the study videos. Has everybody seen the study videos from Pearson? Yes, you know what I'm talking about? Hopefully. Okay, if you have no idea what the study videos are, make sure you ask me or ask Anastasia or ask Ray about it, because they're very, very useful. And have you seen also the study guides from the Pearson? They're very, very useful too. Okay, so what is happening here? First thing that is happening here when you start this whole process of stopping the bleed, right? First thing that happens is as soon as your blood vessel has a little tear, although the angle of this tear is kind of strange, and it's kind of big in comparison to the rest of the blood vessel. So let's say you have a tiny, tiny little tear over here. The first thing that happens is that you're going to have vasoconstriction. Your blood vessel is going to squeeze, okay? It's going to squeeze, like in the middle of this is showing over here. It's like the middle is getting squeezed, okay? And this is essentially called vascular spasm, okay? And what it does, it slows down the flow of blood so that you don't bleed a lot, right? Also, keep in mind that when you have some type of damage to a certain blood vessel, let's say you just hit, like actually happened to me this week. I don't know why, always on my elbow, I'm doing things and I hit my elbow. It was so bad. You know what they call the funny bone? Not funny, right? I hit it so badly. I'm like, okay, I'm just gonna shake it off, but it was hurting and I was gonna get a bruise. I was like, okay, so I went to get some ice and I put some pressure on it, and it took some time for the thing to stop hurting. But I did not, just a little bit of a bruise, but not much, right? But yeah, you put some pressure, and putting the pressure and of course putting some cold compress, you are helping the vasoconstriction to slow down the loss of blood in the area. So that's the first stage that happens, and is very quick. So as soon as you get the tear, your vasoconstriction keeps on happening for a few minutes after it feels that the blood vessel is damaged. Okay. Now the second thing that happens is the activation of your platelet plug. Okay, so the activation of the platelet plug has actually these 3 stages. Right. So the first stage is that collagen fibers are going to interact with these little purple things here. Those are your von Willebrand factors. Okay, those little purple things. And actually here, the purple things are actually the platelets. So it is not showing here the yellow things, actually the von Willebrand factor. Von Willebrand factor is going to get stuck here to collagen fibers, and they're going to call on your platelets to go there and start aggregating. Okay, so von Willebrand is the yellow thing, not purple. Okay, so let's do it here. The von Willebrand factor. Okay. So these little factors are going to serve like glue to your platelets. It's going to start activating the platelet. Okay, so as soon as they bind here to the platelet shifts from being a little, spherical shape to being this star-like activated kind of thing. And notice that the platelet is going to dump out some of those contents that it has in the granules, right? Remember the contents here, we mentioned ADP is right here, and serotonin, right? So those are going to help aggregation of more platelets to this area, and that's your positive feedback, okay? So then when you get enough platelets activated and in the area, you finally get your aggregation of platelet, and that is your plug, okay? So now this is the description here. Then after this is when you're going to start to get the coagulation. So this is the next page here. So coagulation, what is going on here? Actually, to get the third stage here of the blood clot, you have two pathways. One pathway is called intrinsic. So here, the intrinsic pathway, you can have coagulation without a tear in the blood vessel. So things that are inside of the blood can trigger coagulation. These things that are inside of the blood, they have to be negatively charged. Okay, so you always have a little bit of trigger from these intrinsic factors, but then the question is, why is it that you don't get coagulation all the time? Well, because you also have anticoagulant factors that are acting to slow down that spontaneous coagulation. Okay, so you have a balance between procoagulant and anticoagulant, and if you don't have a problem anywhere, so you don't get coagulation. Okay, all right, so if you have factors that are inside of the blood and you're not really exposing anything to the outside of the blood, you call those intrinsic factors, right? This process of coagulation is slower than the process that is called extrinsic pathway. Okay, so when we say we had a little blood tear in the blood vessel, and then you expose your collagen fibers, this is what we call the extrinsic pathway. Okay. So something was exposed from the outside of the blood vessel to the inside of the blood vessel, and this is when you get your von Willebrand factor to go there and serve as a glue to gather your platelets. All right. So extrinsic factor, intrinsic factor, and then you have a lot of factors that get together, and you get your whole cascade going on, and both of these pathways are going to merge in this factor 10 activation. All right, so the merging point is factor 10 activation of the two pathways. And then what happens? This is when you have the activation of thrombin. So you have prothrombin that gets activated into thrombin. And then what is thrombin going to do? Thrombin is going to get fibrinogen that is inactive and is going to make fibrin out of it. Fibrin is going to be part of your blood clot, right? So the whole monstrous coagulation cascade is really reduced for these few steps here, okay? After you get the fibrin, after you have this strong plug on top of your platelets, you still have two other processes that have to happen in order for homeostasis to be reestablished. So after the plug, you're going to have clot retraction. Okay, so this is when your platelets are going to bring the tissue here of your blood vessel together. Okay, so it's clot retraction. And then after that, when you repair your blood vessel, after that, you don't need the plug anymore. So you want to break down the fibrin clot. You want to disperse, all the platelets that got together over there. So in order for that to happen, you go through this last process that is called fibrinolysis. Okay, so you dissolve the clot and everything goes back to normal. Everybody cool with that? Yeah, yeah. All right. So here, clot retraction. What is happening over here? Now, at this space where you have the tear, you have those platelets that are stuck in there. So here you zoom, this figure here is zooming inside a little platelet. Okay, so inside the platelet you have this little cytoskeleton arrangement, and in the cytoskeleton part of this you have actin fibers, you have myosin fibers, and those are going to contract just like you're having muscle contraction. They're going to bring those fibers together, right? So you're going to contract those fibers, and when the platelets contract, since they're stuck to the edges of this little tear, they're going to bring those the sides of the blood vessel close together so that they can undergo repair. Okay? And macrophages are important, or immune system cells are important for repair of those blood vessels, right? So here you have your actin and myosin fibers that are contracting. And when is this taking place? Well, basically within an hour of that little tear in the blood vessel, okay? Now, while the platelets are contracting, they are also putting out healing factors, right? So platelet-derived factors are going to help with the repair of the blood vessel. And so here's a good one that is easy to remember about platelet-derived factor, and this is platelet-derived growth factor. Okay, is growth of the blood vessels components to repair itself. All right, so very, very, very important growth factor for repair. Okay, and once the vessel is repaired, you go through the fibrinolysis, breaking down the fiber plug. Okay, so what is going on over here? You have a certain enzyme floating around in the zymogen form, in the inactive form. Okay, so this enzyme is plasminogen. When it gets to the blood clot, the enzyme is going to be activated into the active form here, which is plasmin. Okay, so plasmin is what is going to dissolve the fibrin clot. All right, so plasmin here dissolves the clot, and everything goes back to homeostasis. Oh, this I have repeated here. I should have taken out that slide. Okay. Okay, everybody cool with what's happening here? All right, so here are the 4 phases of hemostasis. Make sure you study that and you know what is the sequence and what is happening, the important parts that are happening in each one of these phases. All right. So here are some clinical correlations that are interesting, right? One thing that can happen is spontaneous coagulation that can form in case you don't have a tear in the blood vessels, right? So when you have a formation of the blood clot and no apparent reason for that to form, you call that a thrombosis, right? Now, thrombosis is the formation of the blood clot, and then the blood clot that is going to be going through the blood vessels. Now the clot itself, okay, the blood clot itself, you call that a thrombus. Okay, that's a thrombus. Now the thrombus is an embolus. Okay, embolus is something that is floating around in the blood. It could be made of a blood clot, or it could be something else that is floating around in the blood that shouldn't be there. Okay, so embolus is something floating around in the blood. So if it is made up of a blood clot, you call that a thrombus. Okay, so it can be around. When the thrombus or something else floating in the blood clogs up a blood vessel and does not allow the blood to be delivered after that clot, you call that embolism. Okay. So embolism is actually very dangerous because certain organs, or actually all of your organs need blood to survive, right? But certain organs are very, very sensitive to diminished levels of oxygen, right? So you imagine that your brain needs oxygen all the time. If you do not have oxygen delivered to your brain, then you're going to have brain infarct, right? And your lungs also need to have a continuous blood flow, otherwise you don't oxygenate your blood, right? So you have certain organs that are really more sensitive to these problems, to this embolism. Okay. Now there are some health risk factors that put a person that makes a person more prone to developing embolism. One of them is atherosclerosis. And what causes atherosclerosis? Anybody heard of atherosclerosis? Want to see a bit more of this when you get to the blood vessels? Yes. Oh, what? Oh, that's the misconception. I'm glad you mentioned it. Oh, I didn't bring chocolate. I have to remember the chocolate. Yeah, the one thing that everybody thinks about is cholesterol. I gotta stop recording this. It's not really the true enemy. All right, so coming back here, let's go on with more things that you should know, right? So how do you avoid blood clots, especially in a patient population that is prone to developing spontaneous blood clots? Well, older people, usually if they have, especially if they have been in the standard American diet throughout their whole lives, standard American diet, right? If they're sad their whole lives, they usually have some kind of cardiovascular issues, so the doctors will put them in a baby dosage of aspirin, right? In a small dosage of aspirin. Well, why? Oh, because aspirin is going to block one important factor that is coming out of the platelets. So 3 factors important coming out of the platelets. Remember, you have the ADP, you have serotonin, and you have thromboxane A2, right? Aspirin blocks thromboxane A2. So that will prevent aggregation of platelets to form the platelet plug. So baby aspirin dosage is preventing formation of the platelet plug, spontaneous formation. Right? Another factor that is also present in the blood vessels but can be given by injection is heparin. Okay? What heparin does is going to block the formation of thrombin, right? So if you do not have thrombin, you cannot get fibrin out of fibrinogen, right? So heparin is gonna avoid the formation of fibrin, so the third stage of the blood clot. Okay, so heparin is like injection. Besides those 2 factors, you also have this guy over here, warfarin. Okay, it's also called Coumadin. Right, so this is oral medication. There are also people who have either heart problem or atherosclerotic problem. They'll take dosage of Coumadin every day. What this thing does is going to block the action of vitamin K, information of those factors that are important for the coagulation cascade. Okay. Now, this is interesting because you get vitamin K from the microbiome in your gut, but you also get it from dark green vegetables, right? Those dark green vegetables are really going to feed the bacteria to produce your vitamin K. So people who are taking warfarin, they usually avoid taking a lot of dark green vegetables like collard greens or spinach, right? Dark greens, because if you take a lot of those, then they're going to increase the production of vitamin K, which really slows down the effectiveness of the drug, right? So you might know older people who are taking Coumadin for avoiding spontaneous coagulation processes. Oh, all right. So here is your first iClicker question. I have several of them. Hopefully I have time for all of them here. All right, so An injectable heparin medication might be prescribed for a patient who what? Everything else is telling you that you already have a problem making your blood clot, right? So heparin prevents blood clot formation. If you give an injection of heparin to a patient, you do not want blood clot to be formed. Now, if you give heparin to a patient that already has low platelet count, the patient is already not making a blood clot. Now you can have some serious problems because you're inhibiting blood clot in a patient that already has low platelets. You can cause some internal bleeding and the patient might die. Those are serious drugs. Okay? Patient is a hemophiliac. The patient already doesn't have the important factors for a blood clot. You give an anticoagulant, patient may die. Okay, now, deficiency in clotting factor, that's the whole idea. Either the clotting factor or low platelet count, patient cannot make enough blood clots. All right, so here's a fun slide, okay, on actions of vitamin K, all right? Warfarin is going to block the formation of vitamin K from precursors. Okay, so vitamin K is blocked. If you do not make vitamin K, you cannot make 4 of your clotting factors. That means no coagulation is going to be happening, right? And of course, the poison is always in the dosage, right? Warfarin had been used for a very long time as a rat poison. That's why I put this little rat warrior over here, like for warfarin, right? How is it that it causes death of rats? If they eat a lot of this, they're gonna die from internal bleeding. I think it's a bit cruel. I actually prefer those quick rat traps, just like hit the head, quick death. Yeah, choose your mode. All right, so finally, let's get here to the blood types quickly. This is, you probably have seen in another class, but just to reiterate what is happening here with the blood. Do we have, before I jump into the blood type, any questions about coagulation, anticoagulants and stuff? I love to ask questions about anticoagulants in the exam. So make sure you understand the mechanism of how that's happening. Okay, all right. So here, blood types. They're related to issues related to blood transfusion, right? What is a blood transfusion? When you lose blood or when you have some problem with your blood and you need to get blood from somebody else, right? And coincidentally, this morning there was a big red bus right in front of parking lot 1. There's like donate blood today, and they give you, I don't know, tickets to some kind of basketball game or something like that. Or sometimes they give you like $20 or something like that. Right? So basically what happens, you donate the blood, hopefully you're not going to faint when they, I always have that issue, like put the needle in my vein, like, oh, okay. One of the reflexes. Right? All right, donate the blood, and the blood is going to be fractionated. Okay, they're going to get some red blood cells in one section. They are going to get plasma and platelets and white blood cells in another section. Okay, so when you do fractionation of the blood, you get the red blood cells, they are dense, and then you get the plasma on the other side. If you take the platelets and clotting factors out of the plasma, you end up with the serum. Right? So serum is basically just the watery part of the blood with floating proteins like antibodies and other proteins. Okay. All right. So what are antibodies? Right. Antibodies is a reaction of the immune system that we're going to see when we get to the immune system. Basically, you develop antibodies after your body has seen a certain type of antigen. Okay. So antigen exposure is going to allow your adaptive immune system to make antibodies. The cells that make antibodies are your plasma cells that come from your B cells. Okay, so what happens here? In order to make antibodies, you have to be exposed to the antigen. That means your body has to be primed. If you are never exposed to the antigen, you are not going to make antibodies against that antigen. Okay? If the body doesn't see it, you cannot really teach your immune system to make antibodies. Okay? What are antigens? Anything can be an antigen. It can be a little piece of protein. It can be some form of carbohydrates that are chained together. So you can also have carbohydrates, more proteins. They call those glycoproteins. You can have proteins and fats or lipids. You call those proteolipids. So anything that your body doesn't naturally make it can be an antigen. Okay. All right. Sometimes your body makes some things that are seen as antigens. So sometimes your immune system will attack parts of your body, and those are autoantigens or something, or autoantibodies or something like that, right? All right, so let's see what happens here. When you make antibodies, the antibodies have this little Y shape, okay, like this, like heavy chains and light chains, right? So one piece of that antibody is going to bind to an antigen, and the other side of the antibody is going to bind to the same antigen. Okay, so why are we talking about antigens? Well, because our red blood cells, right? Our red blood cells, they have on their surface some sugar molecules that can be antigens. Okay, so here you have a little sugar molecule that can be antigen. Now it depends on which blood you have. So let's say if you have blood type A, then the antigen on the surface is type A. Those little sugars on the surface of the blood cells, those are type A antigens. Now, let's say that you have, instead of type A blood, let's say that you have type B blood. Okay? So your antigens here on your red blood cells, those are type B antigens. Right? So let's say here you have a person that has type A, and you have a person that has type B. Okay, so we have your two people over here. Happy people. Okay. So what's going to happen? Your B person does not have a antigen. Your B person is going to make antibodies against type A antigen. Okay, so your B person is going to make this antibody molecule that is anti-A. It's going to bind to A. All right? Your A person is going to make what kind of antibody? It's going to make anti-B antibody. So this is going to be anti-B antibody. Okay. And we forgot the third person here. The person that does not have any antigen on the surface of the red blood cells, you call that person what kind? O. So this type O person is going to have antibodies against what? Anti-A and anti-B. Okay? The O person is going to have both types of antibodies against both antigens because the red blood cell does not have those antigens. So the person is going to make the antibody against those antigens. Now, I just mentioned that in order for you to produce an antibody, you have to be primed with the antigen, right? And look at this. Okay, Vaccination, okay, routines, okay, this something else. Okay, let me see, I think is the next is right here. Okay, here we go. Anti-A or anti-B antibodies, they're gonna find them in the blood of a baby after 2 months of age. And you continue to build those antibodies up to a regular level of an adult. This is gonna happen before you're 10 years old. How did the baby get primed by the antigens? This is when everybody goes, what is she talking about? Right? I was very confused until a few years ago with this question. How is the baby going to develop the antibodies if they never seen the other blood type? The body was not primed with the other blood types. From the mother, like how? Huh? Breastfeeding. Answering in simple terms, no. But that's a more complicated question if you really think about it. Okay, but no, it's not through breastfeeding. So let's say the mother is type A and the baby is type A. Okay, that was actually a painful question. Imagine if your mother is type B and the baby is type A, and then the baby is breastfeeding. You know, when the baby starts to get teeth and then you go breastfeeding, exposure to the mouth. Okay, it raised that. Okay. No, that's not how you get exposed to the different blood type. Yeah. Huh? Vaccination. That's a good idea of developing antibodies. So the doctors take your baby as soon as the baby is born. They take the baby to a secret compartment. They get different blood types and they inject your baby with all the different blood types. No. I hope not. That's not what happens in this case, no. Another guess. Yeah. Gene expression, no. Normal birth? That has to do with it, yeah. Okay, let's take that. How would that interfere? How would that contribute? Oh, you were thinking about blood explosion. No, I was thinking that you were thinking about all the germs involved in regular birth. Yeah. No, not the blood. Okay, I'm gonna tell you. Okay, I'm gonna tell you. When you mentioned regular, normal birth, I was thinking that you were thinking about all the microbiome that the baby is exposed to through the birth canal until it comes out covered with all these nice germs. Yes, no, that's actually what happens, and even with C-section, the baby is going to come out of that environment and is going to be exposed to all these beautiful bugs that are part of the microbiome. Microbiome is going to get into the intestine. This is when breastfeeding helps, because components in the breast milk are going to feed the good bacteria in the intestines of the baby. And the microbiome is going to teach your immune system to develop. Okay? So from 2 months of age or so, you start to develop those antibodies, because some bacteria in the intestines, they have the same type of antigens as you see on the surface of red blood cells. Okay, so your body is going to think, well, if I see that antigen and I don't have that antigen, that thing is foreign. I'm going to develop antibodies against it. If I see that antigen and I have that antigen, so I'm not going to develop antibodies because that thing is self. Okay? So your microbiome is going to allow the development of these antibodies early in life, and you keep the antibodies, you keep producing antibodies for the rest of your life. The type of antibodies that are produced because of the microbiome interaction, and the type of antibodies that are against A or against B blood factors Those are the type of antibodies that are kind of bulky. They are big. They do not cross the placenta. Yes. They are not going to have anti-A or anti-B because both of those are self. So they're not going to make antibodies against them. Okay? All right. And then somebody who is type O, they're going to make both types of antibodies because they don't have any of those antigens. All right. So then what happened? You produce the antibodies. Antibodies are going to bind if the person receives the wrong blood type. Okay. So antibodies are going to bind one side to one red blood cell. Right to here. Oh, hopefully my pen is going to work. One side to one red blood cell. And another antibody can go over here and bind to that red blood cell, and another antibody is binding to another red blood cell that binds to a different antibody that can bind to another blood cell. And you get a big clump that forms here. The whole clump of antibodies and red blood cells, you call that agglutination. Okay? If you get agglutination happening in case of wrong blood transfusion, these clumps can plug very small blood vessels, and you can cause a lot of problem. And especially because these red blood cells are going to be tagged for destruction, and that can cause even more health problems and even death. Okay? So agglutination due to the wrong type of blood transfusion is life-threatening. Okay? All right. So here are your blood types and the type of antibodies that are formed. Notice that these antibodies are bulky. Those are M type of antibodies, and they are so bulky they do not cross the placenta. Okay? So moms cannot give this type of antibodies to the baby. The baby has to develop their own antibodies after birth. All right, so here's from your book with a little chart, anemia of the baby. I thought I had a slide for that. Oh no, okay. How do you avoid this problem? During the first pregnancy, before priming of the mother's blood, you can block the antigens using this drug that is called Rogam. Okay? Rogam, what it is actually an antibody, only the protein, that you give to the mother that is going to block or neutralize any of the red blood cells of the baby that come in contact with the blood, with the mother's blood. Okay, so you give this drug to the mother, and you prevent the exposure or the priming of the mother. So the mother is not going to develop cells that can produce antibodies. Then for the next pregnancy, the mother did not produce the antibodies, so the baby is safe and you're not going to have hemolytic disease of the fetus. Yes? Whether you have the antigen depends on what you're talking about. Okay, no. What determines is the presence of bacteria in your intestines. Everybody has them. There is no way of avoiding bacteria in the intestine. Yes, Rh factor is hereditary. Yes. So if you have this Rh protein on the surface of red blood cell, then you are Rh positive. If you don't, then you are Rh negative. Okay? And this is very important to always look at the Rh factor when you go through blood transfusion, because you can look at compatible blood, but if you are giving Rh positive to someone who is Rh negative, that's a problem because you're going to be priming them. Or even worse, if the person was already primed before, now you're going to cause death. Okay. Yes, I had a question here. No, okay. Yeah? Is it possible for twins to be born with different blood types? Yes. Unless they are identical twins from the same egg, then they have the same blood type. But if they are fraternal twins, then they can have different blood types. Yeah. All right, so here is the next question. Which antibodies are naturally present in somebody who is B positive? Okay, you don't need more than 30 seconds for this one. It's easy question. Anybody need more time? All right, what is your answer? A. Right. Okay. So what happens if you get the wrong blood? You're going to get those clogs, the agglutination. They're going to clog your small blood vessels. Your red blood cells are going to burst, the ones that were bound by the antibodies, all right? That is going to release iron and hemoglobin out of your red blood cells. Those are very damaging to your kidneys. You can end up with kidney failure, okay? Besides, clogging up your blood vessels is going to obstruct the passage of blood to vital organs. So it's a very serious thing if you give somebody the wrong blood type. Here I put a link for a movie of even a more rare type of blood. So if you have nothing to do on a Saturday night and want to watch a cool movie, a good documentary, I would click on that link. All right, finally, the heart. Chapter 18. Let's dive through the heart here. All right, so I have a few minutes, okay. All right, circulatory system, the heart. The heart is the pump, right? Pump that is pumping the blood. It is small, it's about the size of your fist, closed fist, right? How often does it pump? Well, it depends whether you exercise or not, but in average, about 100 times a day. That's a lot of pumping, right? All right, now, what is it that the blood is doing? Of course, it is moving the heart is really moving your blood around. It's going to carry oxygen, carbon dioxide, nutrients, and waste products, and it's going to allow for, of course, circulation to happen, okay? So here, where is the heart? Right behind your sternum, right, your sternal bone, and is in the middle, in the mediastinum, the middle line, okay? And important things to know here, the heart is sitting right on top of your diaphragm, and that is your very important breathing muscle. Right, so here's your diaphragm. And it is between your second rib, right, second rib and your fifth rib, if I remember correctly here. I think it should be the fifth rib. Yeah, so here. We go 2, 3, 4, 5, right there, around there. So that is your location of your heart. Now, certain characteristics of the heart. You have the top kind of flat side of the heart that is called your base. The tip of the heart is pointing down, and that's your apex. In most people, the apex points a little bit to the left side. Okay. There are some people that have all the organs inverted to the other side. It's kind of rare, but it could happen. Okay, so here, if you are a typical person with the heart, with the apex pointing to the right, to the right side, which is the left. To the correct side that is at the left side, right? Then you're going to find that you have the apical impulse of the heart that is right there, right under your left side, on top of the diaphragm. Okay. Okay, more points. Tell you, full of points today. The apex of the heart points where? Now there's some answer. I'm just trying to imagine how the heart is pointing for these people who are giving me the wrong answer. Hey, consult your friends. Consult your notes. Anybody need more time? Yeah, what is your answer? C. Okay, some people answer me posteriorly towards the left shoulder, so that heart is upside down. Okay, it's not supposed to be upside down. Okay. All right, so we see where the heart is and the shape of the heart, the base and the apex. And now we're going to look at the membranes around the heart. Okay, so the membranes, you call that pericardium. Okay, so the pericardium is right close to the heart, is a double membrane. Okay, they're actually like three levels of membranes here. Okay, so let me see here. I think I have a better diagram for this. Okay, let me go to the next slide here. Okay, so here, pericardium is this membrane that surrounds the heart like a bag. Okay, so here you see the heart has its own surrounding membrane for protection. If you compare with the lungs, each lung has its own membrane sac as well. Okay. So let's see what is happening here. The heart is really totally enclosed by this membrane composition here. Okay, so let's go here. All right. So first level of this membrane that goes around the heart is a very tough outer layer of this pericardium. You call that the fibrous pericardium. Okay, it's very, very dense and not quite flexible type of connective tissue that keeps the heart in place and protects it from pushing of other things. Okay, so the very outmost part is the fibrous pericardium. Okay. So top layer here, so you see here, this is the fibrous pericardium. Right under the fibrous pericardium, you're going to have this double sac layer. Okay, so this double sac here, you have the part that connects with this fibrous pericardium, and this part here is called the parietal layer of the serous pericardium. So here comes your other level of pericardium. Top one is the tough one, is the fibrous one, and this double one that is under the fibrous and close to the heart, that is called serous pericardium. Okay, two layers of that serous pericardium, right? The part of the serous pericardium that connects with the fibrous layer, that is your parietal. The part of the pericardium that connects with your heart muscle, that is called visceral. So when you talk about membranes, the part that always touches an organ, you call that part visceral side. Okay? And the part that is away from the organ and touches the walls of the body or something else, you call that part parietal side. So important terms here: visceral pericardium, parietal pericardium. Okay. Now your visceral pericardium has another name. You also call that the epicardium. Epicardium. Okay. It's very much connected, or is attached. It is touching the muscle part of the heart. Okay. The muscular part of the heart, you call that the myocardium. Okay. So here, just looking at the pericardium, right? So tough part, and then this sandwich type of membranes here. These are the serous part. Now inside this opening section of the serous pericardium, you have some fluid, some serum that fills up that space. Okay, which is not a big space, it's not a lot of fluid, but it's very, very important because, as we all know, the heart is beating all day long. It never takes a break, right? What happens if your heart takes a break? You die, right? So you don't want your heart to take a break, right? And because it's always moving, it can create heat and friction around itself. So this liquid and this pericardium, together with the pericardium, this is buffering and is really avoiding this problem that would be of friction and increased heat and all this other this stuff in case you don't have this buffering session. Okay, so pericardium, this serous pericardium is very important for that buffering function. Okay. All right, so let me see here. I mentioned this, I mentioned that. Okay. All right, so here looking at the heart wall, let's look at the heart wall. So the visceral part of the pericardium is part of your heart wall. Okay, so you have a membrane there, you have muscle here, and you have membrane on the inside of the heart. The outside part is of the membrane here. This is part of your pericardium. Pericardium is around the heart on the outside, right? Then the middle part, you have your myocardium, that is your muscle, and then the inner part, you have your endocardium. So these three parts here, they really make up your heart wall. Okay, so this is the inside of the heart. It goes to the chambers. All right, so this picture was taken from here for the base of the heart. Okay, so here just showing you exactly what I had mentioned before about the heart wall. Okay, so you have your pericardium, okay, epicardium. Now the pericardium here is not giving you specifics on those three layers. So you have your epicardium, which is the visceral part. You have myocardium, and inside here, inside of the chamber, contouring all the surface of the inside of the chambers, you have your endocardium. Okay, another thing about the endocardium is made up of epithelial cells, and it can also be called endothelium. Okay. All right, so here another view of your heart wall. Okay, I mentioned that. So here, chambers of the heart. You have 4 chambers. You have 2 upper chambers. You call those the atria. So you have your right atria that is moving deoxygenated blood. You have your left atrium that is going to move oxygenated blood. Okay. And then you have your lower chambers, and those are your ventricles. Now look at the ventricles. The one that is very beefy here, that has a lot of heart muscle, that is your left ventricle. Okay, and the reason why this muscle is very beefy is that you need a lot of push for the blood that is going to come out of there, because the blood that is oxygenated blood that's going to come out of your left ventricle is the blood that is going to go to the aorta and to your systemic circulation. So circulation to the rest of your body, that is pushed by your left ventricle. Okay, your right ventricle doesn't need a lot of beef because it's going to push the deoxygenated blood to go towards your lungs to get oxygenated. It's kind of like a short path, you know, doesn't need a lot of push, right? All right. So now we divide the heart into 2 hearts. So you have your left heart and you have your right heart. So your left heart is going to receive oxygenated blood from the veins that are coming from your lungs. So now this is another term that is important to know. If blood vessels are arriving at the heart, you call those veins. So blood vessels that arrive to the heart, those are veins. So it doesn't matter that they are coming to the right side or to the left side. If they're arriving at the heart, they are veins. If blood vessels are taking blood away from the heart, so getting the blood away from the heart, you call those arteries. Right? So the artery here that is coming from your left ventricle, and this big artery that will distribute the blood for the rest of the body, that is your aorta. Okay, aorta artery. The artery that is coming from your right ventricle and is going to push the blood here, is going to take the blood to your lungs. You call this your pulmonary arteries. Okay, so artery here, pulmonary trunk, and pulmonary arteries. Okay, so arteries go away from the heart, take the blood away from the heart, and veins bring the blood back to the heart. So your left heart is going to pump the blood to your systemic circulation. Your right heart is going to pump the blood to your lungs. It's your pulmonary circulation. Okay. So we see before, your left side of the heart is the beefy one, so that has to be your strong pump. Okay. The right side of the heart is the not so beefy one, so that is your relatively weak pump. Okay. Now, besides left and heart, you also have other pumps that can be considered strong or weak in relation to each other, meaning that when you look at the atria of the heart, at the top of the heart, those are also relatively weak when you compare it to the ventricles, right? So the top of the heart, meaning the left and the right atria, those are considered weak pumps, and actually the atria, they don't really have to pump a lot, because the pumping of the atria doesn't really contribute a lot for moving of the blood circulation. Okay. So your bottom heart is actually your strong pump, and the bottom left is even stronger, right, than the bottom right. Okay. So right side of the heart, right ventricle is going to coordinate blood to your pulmonary circulation, and left side of the heart is going to circulate blood to your systemic circulation. Okay. All right, so here is that information on the weak pumping of your atria, right? So basically, when blood arrives at the atria, let's look here at your right atrium, right? When blood gets there, it will fill up the atrium, and then it is going to create enough pressure here that is going to open the valve that is between the atria and the ventricle. And when that happens, then the blood will just drop to the bottom part of the heart. Basically, it will just drop there because of gravity or inertia. It will just, the valve opens, the blood drops. So after the blood drops, you're going to have the atria that are going to contract, but you don't really need that contraction for your blood to get to the ventricles. All right. So the contraction of the atria, you call that really just the atrial kick. It's like the blood dropped and you give it a little more push, and then the blood finally gets to the ventricle. Now, if your atria for some reason do not work, it's okay, you can survive without a problem, because you don't really need the atrial kick for the rest of your heart to work. Okay, with the atrial contraction, you only get an increase of 20% of the blood that is pushed to the ventricles. All right, so somebody can survive without atrial contraction, basically without a lot of trouble. Okay. All right. So what controls the direction of the blood is basically the difference in pressures, and what builds up the pressure is really the presence of the valves. Okay, so you have two sets of valves. You have valves that are between the atria and the ventricles. Those are called your atrioventricular valves. And then you have valves that are on the outflow part of the heart. So you have a valve that is right there at the entrance to your pulmonary trunk, and another valve that is right there at the entrance to the aorta. So those are your outflow valves. They are also called semilunar valves. Okay? Yes, I know I am blowing up the time, so we're going to stop right here, and we'll pick up from here next class.